Here on part 5, we're going to talk about the nitrogen cycle. Archaea and cyanobacteria changed the world in terms of an additional reaction. That additional reaction is the nitrogenase reaction, in which archaea and cyanobacteria are able to take diatomic nitrogen and reduce it to ammonia. The energy within those nitrogen-hydrogen bonds can then be used as an energy source for different metabolic processes. This environment, this reaction evolved in prokaryotes. It does not exist in eukaryotes. Eukaryotes, as we'll discover in a few lectures, have the capacity to form a symbiosis or long-term relationship with a prokaryote. But eukaryotes themselves do not conduct this reaction. Only cyanobacteria and archaea perform that reaction. The nitrogenase reaction takes diatomic nitrogen and reduces it to produce ammonia. It takes a large number of protons and electrons and a very large amount of energy to break apart the diatomic nitrogen and to store the energy within the nitrogen-hydrogen bonds. This reaction of nitrogenase evolved in the world in which oxygen was not present. And it turns out, just as with rubisco, nitrogenase is oxygen sensitive. The nitrogen cycle is a series of reactions associated with taking diatomic nitrogen to form ammonia, and then through a series of adding oxygen or taking away oxygen, we transform ammonia to nitrite, to nitrate, to nitric oxide, to nitrous oxide, and back to nitrogen. The process of taking diatomic nitrogen to create ammonia is called fixation. The process of adding oxygen to the nitrogen is called nitrification. And the process of removing oxygen is known as denitrification. There's a fourth process, which is the process of taking the protein, which is in plants and animals, breaking it down into its amino acids, and then converting those amino acids back to the ammonia form. And that's known as mineralization. Early on, this entire nitrogen cycle was conducted by prokaryotes alone. And here we see diatomic nitrogen coming into the organism through a nitrogenase, nitrogen fixation reaction. And then ammonia can then be used to make proteins, or it's nitrified or denitrified through a series of reactions within microbes back to diatomic nitrogen. This is a cycle. The cycle involves only archaea and bacteria. It does not involve eukaryote, whether they're algae or higher plants. So early on, this was the nitrogen cycle. When we add plants to the system, what we discover is that plants have the capacity to take up ammonia. When they take up that ammonia, hydrate it to ammonium, it's used to make amino acids, which are then used to synthesize proteins. Or the plants can take up nitrate and then reduce that nitrate to ammonia using a nitrogen reductase. And then that ammonia is used to make amino acids and eventually proteins. The other part to complete the cycle in plants is that when a plant or animal dies, that protein is released into the environment, broken down to amino acids, and then ammonification or mineralization takes place where that ammonium, the amide group, is then converted to an ammonium. In a low CO2, a low oxygen world, all cells of cyanobacteria, such as this Cyanobina, had the capacity to fix nitrogen. But because this enzyme turns out to be sensitive to oxygen, what we find is that these single-celled organisms come together to form a community, with some of the cells specializing. They specialize to create a cyst with a low oxygen environment. That low oxygen environment allows the nitrogenase reaction to take place. So within that cyst, we find ammonia production, and that ammonia is released and then made available to other members of the community. In this symbiotic relationship, the energy for the nitrogenase reaction to take place comes from the autotrophic or photosynthetic cells. So we have a community in which some cells provide the carbohydrate, other cells provide the nitrogen, and the result of this is energy for growth through carbohydrates and ammonia for protein synthesis through the nitrogenase reaction. Obviously, there is signaling that takes place because the carbohydrates have to transfer to the cyst and the ammonia needs to transfer to the cells. We find that the symbiotic relationship can actually also take place in higher plants, where a higher plant, a eukaryote, is not able to conduct the nitrogenase reaction, but a bacteria, a free-living bacteria, or a symbiotic bacteria, rhizobium, is able to take diatomic nitrogen and form ammonium. In plants, the plant forms a cyst known as a nodule, and that nodule is able to create a low oxygen environment in which the microbe, in this case the rhizobium, is able to produce ammonia. That ammonia is used for plant growth, and in turn, the plant is supplying the bacteria with carbohydrates for growth and for the nitrogenase reaction.